Hi, my name is Chanèle and I am a PhD candidate in the lab of Dr. Cyr-Depauw at the University of Ottawa. So in our lab, we are interested in finding treatment for neonatal lung diseases by using cell therapy and gene therapy. And my PhD project focused on a disease called bronchopulmonary dysplasia, or BPD. And BPD is the most severe complication of extreme prematurity. It is a chronic lung disease that arises following mechanical ventilation and exposure of a high level of oxygen. And BPD is characterized by an arrest in lung development, where the alveoli are much bigger, and there is less number of alveoli, there is thicker septa and dysmorphic microvasculature, as well as inflammation. An infant that suffers from BPD will develop other lifelong consequences. Some of them will develop pulmonary hypertension, for example. And currently there is no cure for BPD, but previously in our lab we have shown that the injection of MSCs in a hyperoxic neonatal rodent model can improve the lung structure as well as the lung function. So for my PhD, my objective here was to compare the healing effects of UC-MSC donors in hyperoxia-induced experimental BPD. So we hypothesized that MSCs isolated from different donors with various perinatal conditions will have different therapeutic potential. And if you look here at this summary circle, it shows what I've been doing for this study. So we get the cord and we isolate the MSCs from the human cord, and then I use these cells to inject the hyperoxic rat pups, or I use these cells to do single-cell RNA-seq analysis with the goal to identify superior repair therapeutic cells. So here in the method section, there is a little bit more details about the models. So the pups are exposed to 85% oxygen from postnatal day 0 to postnatal day 14. And at P4, the pups are injected either with PBS or the cells. And at P21, we assess different endpoints. In terms of the experimental groups, there is one control group, which are pups kept under normoxic condition, a lung injury group, which is the hyperoxic group injected with PBS, and 5 other lung injury group that are injected with MSCs coming from different donors. So some donors are term donors and other donors are preterm donors, and preterm donors with preeclampsia, preterm donors with chorioamnionitis. So there is a little bit more of information on the different donor in this hyperlink here, and these are all the different donors that we used for the MSCs coming from the different donors that we used for the single-cell RNA-seq. We also used the human neonatal dermal fibroblast as a control. So for the single-cell RNA-seq, we plated these cells in a 96-well plate, and we used the multi-seq technique where every replicate have its own barcode, so that we can pull together all the samples and do one run of a single-cell RNA-seq using the 10x genomics platform. So for the results in the in vivo experiments, we look at the survival, and we see that there was an improvement in the survival of the hyperoxic rat pups when they were injected with the MSCs, but not when they were injected with the MSCs from the term 2 donor. Then we look at the lung structure, and we can see on these images, which are images of the lung section stained with H&E. In the lung injury group injected with PBS, there is much bigger alveoli, thicker septa compared to control, and then there seems to be an improvement in the lung structure of the animals injected with the MSCs, except the T2 MSCs. And this was quantified by mean linear intercept assessment, which is basically a measure of the distance between the alveolar wall. So the MLI was increased in the PBS group, and the MLI was decreased in the in the animals that were injected with the MSCs, but not with the T2 MSC. We also look at the lung function. For this, we use a flexivent machine, which is connected to the lung of the animals via the trachea, and it can apply increasing amount and decreasing amount of pressure to the lung of the animals and measure the lung volume. And then from this, we extrapolate the PV loop, and we see a shift towards the bottom of the PV loop in the long injury group. And there is the PV loop is shifted towards the top when the animals are injected with the cells, but not the T2 MSC. And then we look at the lung volume at the highest pressure point, so the volume at pressure 30, and we see a significant decrease in the lung volume of the animals injected with PBS, and the MSC injection improved the lung volume except the T2 MSC. So then we also look at the right ventricular hypertrophy, knowing that this is a sign of having pulmonary hypertension, which a lot of BPD patients will develop. And we saw that there was a significant increase in the Fulton index in the PBS injected animals, and then the MSCs reduced the Fulton index. This is ongoing work, so I don't have the T2 MSC group here, but yeah. So we also look at the lung vasculature by doing micro CT images. So here we show that in the lung injury PBS group, the vascular volume is smaller in that group compared to the control group, especially in the vessel that are the smallest one. And the injection of MSCs increase the volume of the smaller vessel. So then for the multi-seq single-cell RNA-seq analysis of UC-MSCs, what we have looked at, we look at the cell clusters. So we show that there was 7 different cell clusters that we identified. And here in this graph, we show the distribution of the MSCs from each donor within the different clusters. So most of the MSCs are in cluster 0, but some MSCs coming from some particular donors, they cluster by themselves. For example, the T2 MSCs are mostly into cluster 3. And then the fibroblasts are in cluster 6. And then we look at the ISCT MSC cells, so first markers, and the MSCs express these markers, and they lack the expression of these markers. Then we look at the marker genes per cluster and their enriched coterms. So what we found is that in cluster 6, which is made of which is composed of the human neonatal dermal fibroblast, a lot of the enriched coterm are processes that are related to fibroblasts, such as extracellular matrix organization and all these collagen processes. And when we look at cluster 3, which is the T2 MSCs that didn't have therapeutic effect in our in vivo model, there is also a lot of enriched coterms that are fibroblastic-like. And then in the cluster 0, which contain MSCs that had therapeutic effect in an in vivo model, a lot of the enriched coterms are related to cell signaling functions. So in conclusion, we show that the MSCs from every donor except the T2 MSCs improve the survival, the lung structure, and the function. And MSCs isolated from term and preterm delivery, so the T3 MSCs, P5 MSCs, they attenuated the signs of pulmonary hypertension and increased the lung vasculature volume. And we showed that the majority of UC MSCs, they clustered together, except for some MSCs coming from some donors, such as the T2 MSCs that forms their own cluster and they exhibit different expression patterns. So if you have any question, you can always email me at this email over here. And thank you for listening.